Hi everyone, my name is Dave Evans. I'm from the University of Queensland, and I'm going to be talking in this lecture about some ways that we can extend the classical twin design using structural equation modeling. And we've called this piece of work the Augmented Classical Twin Design, and it's basically a way to use SEM to test the equal environments assumption in twin studies. So in the classical twin design, basically what we do is we compare the trait similarity between MZ twins and DZ twins and then use it to estimate heritability and the proportion of environmental variance due to common environmental factors and unique environmental factors. But the classical twin design rests on a number of assumptions, probably the most important of which is the equal environments assumption. And it basically posits that trait-relevant environmental covariation in MZ pairs is the same as that found within DZ pairs. And violation of this assumption will inflate heritability estimates in classical twin studies. There's kind of a number of caveats to this. So first of all, the environmental factor must affect the trait in order for violation of this assumption to be an issue. I mean, if it's just the fact that MZ twins experience more similar environments than DZ twins, but then those environments don't affect the trait, that's kind of irrelevant when we're estimating heritability for that particular trait. And there certainly is good evidence that MZ twins do often experience more similar environments than DZ twins, but the environment has to affect the trait in order for it to bias estimates of variance components. And the other thing, of course, is that twins have to be kind of passive recipients of these more similar environments for violation of the equal environments assumption to affect the heritability estimates. So the EEA has been hypothesized to be a possible reason for the missing heritability in genome-wide association studies. So remember, the missing heritability problem is this idea that in GWAS, the genome-wide significant variance only responsible for a small amount of the phenotypic variance of the trait, much less than the heritability estimates from twin studies. And kind of one of the thoughts is that, well, maybe heritability estimates from twin studies actually biased upwards because of violation of this equal environments assumption. So over the years, there's been a number of different methods for testing the validity of the equal environments assumption. Probably the easiest thing to do is to measure environmental similarity between twins and then examine whether this correlates with trait similarity as well. Of course, the problem is that you need to measure the relevant environmental factor, and that's not always easy or not always possible to do. There's also been clever methods based on mistaken zygosity diagnosis. So kind of the rationale here is that if parents treat MZ twins more similarly than DZ twins based on some preconceived notion that the MZ twins are more similar than DZ twins, then trait similarity should be a function of perceived zygosity. And this has the advantage that it kind of tests all environmental factors simultaneously. So you don't have to worry about measuring that single relevant environmental factor. The disadvantage, of course, is that it requires, there to be mistaken zygosity diagnosis. So we were interested in whether there could be some ways to use the widespread availability of genome-wide genetic data to investigate this question. And it got us thinking about this paper from 2006 by Peter Vischer and colleagues, where they invented a new method for estimating heritability using identity by descent sharing in sibling pairs. And basically the idea in this paper is that they modeled the phenotypic similarity between full siblings as a function of their genome-wide genetic similarity, in particular their identity by descent sharing across the genome. And the advantage of this approach compared to the classical twin design is that it enables the estimation of trait heritability without many of the assumptions of the classical twin design, in particular, the equal environments assumption. The disadvantage is that it requires large numbers of sibling pairs to estimate heritability precisely. But we were interested in whether we might be able to leverage this idea in order to investigate violations of the EEA in the classical twin design. So I'll just quickly review the concept of identity by descent and identity by state, just to make sure that everyone's kind of on the same page. So identity by descent, we basically say two alleles are IBD, identical by descent, if they are descended from the same ancestral allele. So in this left, this pedigree on the left, you can see that the one allele has come from the common ancestor, the mother here. And so we say that the one allele is identical by descent and identical by state. In contrast, on the pedigree on the right, the one allele has come from the individual's mother in the case of this daughter here. But in daughter number 2, the one alleles actually come from dad. And so we say in this particular example, the one allele is identical by state only. It's not identical by descent. And so there are various algorithms which you can use to work out identity by descent at a single genetic locus, and indeed right across the genome. And so the next couple of slides are just kind of giving you an intuition as to how you do that. So in some cases, it can be relatively trivial. So here, the daughters have got different genotypes, so therefore they must be IBD 0 by definition. In this example, they're going to be IBD 2, and you can kind of see that automatically just by looking at the transmissions from parents to offspring. It's not particularly difficult. Things can get a little bit tricky when you have only partially informative cross at that particular locus. So here, we're not quite sure whether, whilst we know that the one allele must be identical by descent, we're not sure whether the 2 allele is identical by descent or not. So it's 50% chance that the daughters are IBD1 and 50% chance that they're IBD2. And things get even more complicated when you don't have the parental genotypes to help you work out the IBD status. So in this particular example here, whether they're IBD 0, 1, or 2 would actually depend upon the frequency of the one allele in the population. And fortunately, there are various algorithms that will allow you to calculate IBD, both at a single genetic locus and across the genome as well. Okay, so that's kind of a whirlwind tour of IBD. How are we going to use that in our model to investigate the equal environments assumption? So in this slide, I've just put up a path model for the classical twin design that you would have seen in previous lectures in this course. So this is a variance components formulation of the classical twin design, in that we are estimating the variance of the latent A, C, and E factors, and we are setting the path coefficients to 1. And of course, we're also got the covariance between those latent factors as well, because they're not standardized, they're not these double-headed arrows are not correlations, they're actually covariances between the latent factors. So hopefully this is revision from earlier on in the course. On the right-hand side of the slide, you've got the variance covariance matrix both for MZ and DZ twins, and you could derive easily the expected variance and covariance using your path tracing rules that you learned earlier on in the course. So that's the situation with just the generic classical twin design. The idea behind the augmented classical twin design is that we are going to include genome-wide identity by descent information into the DZ twin pairs. So you can see this covariance path between the latent additive genetic factors, where this used to be 0.5 VA, now it's pi hat VA. So pi hat is the genome-wide proportion of alleles shared identical by descent for that particular pair of DZ twins. So pi hat is going to vary across twins, and it is a quantity that you can estimate by certain software programs. So Merlin, for example, is a software program that will enable you to do that. So pi hat varies between 0 and 1. So if pi hat is a 1, that's basically saying that the DZ pair is genetically identical at right across the genome. If pi hat is 0, then it means that the DZ twin pair is unrelated. Of course, we're looking at DZ twin pairs, you're never going to see either of those extremes. Rather, pi hat's going to be round about 0.5, but it's going to vary across twin pairs, but it'll be around that 0.5 value. What you'll see is it'll vary somewhere between about 0.38 and 0.62, depending on the twin pair that you look at. And so kind of the insight of this model is that by including pi hat in the expectation for the DZ pairs, it gives us information about VA and the heritability of the trait. And by doing so, that frees up a degree of freedom to do something else with. And what we're going to do with that extra degree of freedom is we're going to use it to investigate the equal environments assumption. So here, what we've done is we've used it to estimate different variance components for the common and unique environment for MZ and DZ twins. So you notice that those variances for MZ twins, for VC and VE, they're specific for MZ twins. They're different to the common environmental variance and unique environmental variance for DZ twins. Okay, you've got different, completely different parameters there, whereas your VA is constrained to be equal across MZ and DZ twins. And so if you follow your path tracing rules, you will get the expected phenotypic covariance matrix on the right here. So I should say that there's also a constraint in this model as well, and that is that the sum of the common and unique environmental variance components for MZ twins is the same as the sum of the common and unique environmental variance for DZ twins. And so a test of the equal environments assumption is basically whether you can set the common environmental components as between MZ and DZ twins equal, or equally, whether you can set VE equal between MZ and DZ pairs. It's the same thing because of this constraint here. So basically, in summary, by including empirical information on the IBD status of the DZ twin pairs, we've got ourselves an extra degree of freedom, and we're using it to estimate different environmental variance components in MZ and DZ twins. So that was a variance components formulation of our new model. A completely equivalent way to fit the same model is using a path coefficients framework. So you would have seen something similar earlier on in the week where you were comparing the path coefficients formulation of the classical twin model with the variance components formulation. This is the same thing here. So we're setting the variance of the latent factors to 1, and we are estimating the path coefficients of those latent factors. Here, we only have 2 latent factors, one for additive genetics and one for the unique environment. And so we kind of quantify the degree of environmental similarity in the twin pairs by this correlation between the unique environmental, between the environmental factors in the twin pairs. So you could imagine that if REMZ here was equal to 0, this would basically be equivalent to an AE model. There'd be absolutely no common environmental sharing between MZ twins because this was 0. Whereas if this were 1, there'd be no unique environment. Here, E would be just a common environmental factor. I mean, obviously you couldn't have that in reality, but just helps to kind of understand the model intuitively. So this correlation here is telling you how much is due to sort of shared environment and how much is due to unique environment. So it's exactly the same as the previous slide. It's just a different way of parameterizing the model. And so here, in order to parameterize the augmented classical twin design, we've got 2 different correlations, an RE for MZ twins and an RE for DZ twins. And so the test for violation of the equal environments assumption is whether those 2 correlations are different. And so on the right, again, we've got the expected covariance matrices that you can derive by using your rules of path analysis. And so, what we have here is just exactly the same model. It's just a slightly different parameterization of it. To be fair, I find this formulation a lot more easy to understand intuitively why it corresponds to a test of the equal environments assumption. Numerically, though, when it comes to actually fit this model, you can get into problems with it. And so when we fit this model, the augmented classical twin design, we tend to do it in the variance components framework. It's just a bit more friendly numerically, and you can get post hoc estimates on these correlations. You know, the two models, as I said, are mathematically equivalent. Okay, so this graph is just looking at some of the properties of the model. In this case, it's looking at the power to detect violations of the equal environments assumption. So power is on the y-axis. On the x-axis, we've just put up the two different formulations of the model. So either the variance components formulation, so the x-axis is written in terms of the common environmental variance of MZ and DZ twins, but also underneath that is written the equivalent path coefficients formulation. So rather we're looking at, you know, the correlation between the environmental factors in MZ and DZ twins. And so what you can see here in this graph is we're looking at two conditions. We've got 20,000 MZ twins and either 20,000 or 50,000 DZ twins. And we're looking to see how power varies across the different conditions. So what we see, perhaps unexpectedly, not unexpectedly rather, is that power increases with increasing numbers of DZ twins, differences in the degree of environmental similarity between MZ and DZ twins, and increased proportion of variance due to the shared environment. It's also kind of a little bit sobering to look at the numbers involved here. You know, these are quite large departures from the equal environments assumption, and yet we need very large sample sizes in order to detect them. So that is quite sobering. This is a graph just looking at power to detect violations of the equal environments assumption as a function of missing heritability. So if we were interested in the degree to which violation of the EEA might be an explanation for missing heritability, this is a graph that informs on this. And again, it's kind of basically saying that we are going to need very large sample sizes to detect violation of this assumption. And, you know, we can only really do it if it really is explaining large discrepancies in the missing heritability. We also looked at the effect of number of MZ twins on our power to detect violations of the EEA. And this was quite interesting in that our results suggested that the number of MZ twins is actually not all that important in terms of power. It's really the number of DZ twins that's the important consideration. And this does actually make sense if you think about it, because MZ twins, they're kind of only, most of the information they're providing is on the value of VE MZ, which is maybe a less important kind of parameter. I mean, you can estimate VE MZ very, very precisely from a small number of MZ twins. And so just by increasing the number of MZ twins, you're not actually gaining all that much. In contrast, most of the information is coming from DZ twins, because that's the IBD sharing between DZ twins is what you're using to estimate VA. And once you've estimated VA, this then has flow-on effects to the other parameters. So the VCMZ, the VCDZ, et cetera, et cetera. And so it's really the DZ twins that are driving the power in this model. Another thing we looked at was the possibility of including a variance component due to dominance in the model, because as well as indexing IBD sharing to help estimate VA, we can do essentially the same thing to estimate VD, except rather than the pi hat, the proportion of alleles shared identical by descent, now it's the proportion of alleles, I guess it's the proportion of genotypes shared identical by descent. So we're looking at whether the pair shares both their alleles identical by descent and the degree to which this is right across the genome. So you're basically calculating the probability of the pair being IBD2 right across the genome. So that's what that little coefficient is there. And so that will allow you to estimate VD in your model as well as VA. So that's potentially useful, because if dominance genetics is actually contributing to twin similarity, you want to model it as well. And if you don't model it, then that's going to give you spurious evidence for violation of the equal environments assumption. And so we basically showed that you can model this component, and if you do so, it'll bring you back to appropriate type 1 error rates for tests of the equal environment assumption. And in the case of model dominance, it will just reduce your power slightly. So there's probably, nothing to be lost, or not too much to be lost in terms of including this in your model. Like any model that you use, there's going to be assumptions. I've listed some of them on this slide. So the usual assumptions in this world, so that's the absence of gene-environment interaction, covariance, assortative mating. You want bivariate normality for your phenotypes. But the single most important thing, I think, is that any unmodeled factor that increases MZ pair similarity relative to DZ pair similarity is going to produce spurious evidence for violation of the equal environments assumption. So in the case of dominance, we can model that. That's well and good. But there's other things that are much more difficult to model. So for example, if there's other forms of genetic non-additivity, and I'm particularly thinking about epistasis, that's going to be problematic for this model. It's also worth thinking of things like errors in your IBD calculation of DZ pairs. That would be problematic. If you've got gene by age interactions, maybe you want to balance age across MZ and DZ twins. If you've got gene by sex interactions, maybe you only want to do this in same-sex DZ twins. And of course, remember, it's important to model the X chromosome if you're trying to calculate pi hat across the entire genome. So don't forget the X chromosome. Given the large number of twin pairs that this method requires in order to have adequate power, and given that twins can be more difficult to collect than sibling pairs, we also looked at whether there was value in including non-twin siblings into the design. And the short answer is absolutely, yes, that can be very advantageous in terms of increasing power. But of course, I guess you have to take into account the possibility that there might be gene by age effects and things like that. So The inclusion of non-twin siblings might be very useful for some traits like height, if you're looking at height in adults or something like that. But it may be more problematic if you are looking at traits that vary across the lifespan. So in conclusion, the augmented classical twin design can identify violations of the equal environments assumption. It requires very large numbers of DZ pairs for power. The model is extremely sensitive to unmodeled factors that increase MZ compared to DZ similarity. So in particular, things like epistasis. And the inclusion of non-twin siblings in the analysis may improve power, but would limit application of the method because of gene by age interaction concerns. I'd just like to finish by acknowledging my co-authors, in particular Daniel Huang, who was the first author of this study, and Brittany Mitchell. Also, if you're interested in reading more, you can find the study described in Behavior Genetics in a volume from 2021.